When DNA is beginning to replicate, it forms a structure called a DNA bubble. You can see the replication fork of the replication bubble and see that half of the copied strand is continuously made as a leading strand, while the other half is made in fragments and is known as the lagging strand.
Moving on to how specifically DNA is replicated, at each end of the replication bubble there is a replication fork, or which is the Y-shaped region where the parental DNA strands are being unwound. At each replication fork, the strands of the double helix are untwisted and separated to become template strands by enzymes called helicases. In order to keep the parental strands separated, single-strand binding proteins bind to the parental DNA. Additionally, while the helicases unwind the DNA at the fork, enzymes called topoisomerase work ahead of the replication fork and prevent tighter twisting of the strands. To begin new DNA synthesis, an RNA chain called a primer is synthesized by an enzyme called primase. Once this chain is made, then the enzymes that synthesize DNA nucleotides can add on to this pre-existing chain of RNA. The specific enzymes that synthesize the new DNA to the pre-existing chain are called DNA polymerases. Since DNA runs anti-parallel, each strand is given a direction, whether it is from the 5' to 3' end or the 3' to 5' end. Due to the structure of DNA polymerases can only add nucleotides to the free 3' end. Therefore, the strand will always elongate in the 5' to 3' direction. The leading strand, in this case is the top strand of new DNA, is synthesized complementary to the parental strand and continuously in the direction of the replication fork, or going into the fork. The lagging strand, in this case is the bottom strand, must work away from the replication fork because it has to add on to the free 3' end. Due to this, the lagging strand is synthesized discontinuously in what are called Okazaki fragments. Once DNA polymerase 3 forms the fragments, a different DNA polymerase 1, replaces the adjacent RNA nucleotides of the primer with new DNA nucleotides. Once this has been done with the roughly 5 fragments in this example, DNA ligase must bind the final nucleotide of the replacement DNA to the first DNA nucleotide of the neighboring Okazaki fragment. This is because DNA polymerase 1 is unable to join these two fragments together. Overall, the synthesis of the leading and lagging strands are completed at the same time within just a few seconds, despite what their titles may imply.